Hi, and welcome to another edition of Acadia's Brown Bag Seminar Series. My name is Tim Watkins. I'm the Acting Science Coordinator at Acadia National Park, and it's my great pleasure to introduce today's speaker, Chrissy Murphy. She is with the USGS, U.S. Geological Survey, Maine Cooperative Fish and Wildlife Research Unit with an office on the Orono campus of the University of Maine. Chrissy has been connected to Acadia for a little bit. She received a Second Century Stewardship Fellowship from the Schoodic Institute as part of the 2021 cohort. And with that fellowship and her ongoing research, Chrissy has been studying food webs in Acadia National Park's lakes, including how those food webs are linked to terrestrial food webs. And she does all of that by analyzing stable isotopes in fish and other organisms. And I had the pleasure of meeting Chrissy in person earlier this summer, about a month and a half ago, in the pouring rain at the south end of Long Lake, Long Pond. We stood there after she had done a collecting trip with rain dripping down our faces and her wet hair was getting in her eyes and all the rest. And I learned at that point that she's from Oregon, which suddenly made everything, it all made sense that we were there in the rain and happy. And she has discovered and told me that fish communities in these lakes are weird. And so I'm very much looking forward to her presentation. She'll tell us how and why they are weird. So Chrissy, welcome. Thank you so much for taking this opportunity to speak to us, and look forward to hearing what you have to say.
Thanks. Thanks everyone for coming during your lunch hour. Okay, so I'm going to talk quickly about what the Cooperative Research Unit is and what that means, and then about the freshwater food webs of Acadia National Park and some of the linkages to the terrestrial food webs as well. So my email is listed down below. You'll notice it's at maine.edu, and that ties into how the cooperative research units are structured. First, I want to thank Jason Wiedup, and I'll talk a little bit about who he is as the presentation goes along. Allison Jackson, who might be familiar to folks as a former Second Century Steward Fellow as well, and a collaborator on the project, along with Amanda Clemmer, Mike Kinnison, and Angelina, Kristen, and Samantha, who are students from Purchase that worked for UMaine this summer in order to do some of the field sampling that I'm going to talk about. So this wouldn't have been possible without a huge number of collaborators and cooperative partners, and that's really important to keep in mind. I get to stand here and take credit for what is a really great group effort. So the cooperative research units, before we talk about the park in particular, are essentially USGS offices of usually 2, 3, or 4 unit scientists that are embedded in universities across the US. The program is almost 90 years old, and it was designed to develop a conservation workforce through graduate education, fulfill training and technical needs of cooperators, and deliver actionable science. In Maine, the primary cooperators are USGS, the University of Maine, IF&W, the U.S. Fish and Wildlife Service, and the Wildlife Management Institute. And so we function, for all intents and purposes, like university faculty, but we straddle this world of federal, state, and university partnerships. So in the Maine Cooperative Research Unit is myself, Joe Zaleski, who's the unit leader, and we at full capacity have 3 staff members. So that would be another assistant unit leader focused on wildlife. We also have support staff who help us get through the day-to-day, and a tremendous privilege of working with some really excellent graduate students. So that will come up as we move forward. As you heard, I'm from Oregon, so places where it rains, but it doesn't always rain here. This is out field sampling back in June. I started working at a state agency in Oregon and moved on to the Corps of Engineers in Oregon State University. So my experience kind of straddles both worlds. I've conducted research in Oregon, Washington, Idaho, Ecuador, Chile, Spain, and now Maine, which conveniently rhymes. And I work often on stable isotopes as a technique to get at some of the linkages in aquatic ecology between fish and plankton, parasites, things like that. So to give you an example of kind of how we've used food webs and stable isotopes in other areas, in Oregon, we use them to look at reservoir management. So they can be a way to look at how changes to a system will impact different fishes in a community, looking at kind of the who eats who, if you have a taxa of particular interest. So where I worked before, that was Chinook salmon, but they really lend themselves well to some of these applied questions. When we think about freshwater food webs of Acadia National Park, the question that we had coming into this is, does the composition of a fish community predict trophic relationships and cross-system subsidies? And so that cross-system subsidies is a fancy way of saying how things like emergent insects, which actually end up then feeding the terrestrial system, change when the fish in the lake change. And different fish can eat different things, just like you and I might eat different things depending on, you know, what our circumstances are. That's true for fish as well. Acadia is really interesting because of this, and not just because there's already data on mercury, which is a boon for collecting stable isotope data because mercury tends to look a bit like nitrogen. This is from the Dragonfly Mercury project, and the only real thing that I need you to take away from this is when we get to talking about what nitrogen looks like, you'll notice the same kind of stair-step pattern that you see in the middle there, which is that as you know, something eats something else, nitrogen accumulates. Similarly to mercury, but not exactly the same, because mercury is influenced by the habitat characteristics, for example. And when you talk about that aquatic to terrestrial linkage, the strength of that relationship rate is going to depend on how much material is moving. So the reason these lakes are a little weird and really interesting is that they have a history of introductions that's not the same for every lake. So you have this tremendous number of lakes that have a diversity of freshwater fishes that are usually shared among at least a few lakes, but where of 28 species that were recorded in 1999, almost half of them were introduced. So the composition, because they're not all introduced in the same place, is often predominantly native taxa with like one or two new fish that vary depending on where you are. And then there's actually one additional introduced fish since this report, at least one, which is yellow perch. That's pictured there. So it's right now, if all things are equal, you'd still have 15 likely native taxa and 14 that are likely introduced. Some of those are native to other parts of Maine, but people like to move fish around. So for example, here's Jordan Pond, Long Pond, Eagle Lake, and Aunt Betty Pond and the compositions. So when you look at this, the blue fishes are likely native and the brown are likely introduced, and you'll see there's a lot of overlap, and yet it's not perfect. So it really lends itself to asking that question of, if you add a certain taxa, does it start to shift maybe, for example, what a brook trout would eat? Because you can look at systems that have brook trout and that may or may not have yellow perch, for example. So we picked those lakes in order to overlap with some of that prior information. And we focused on food webs because we're interested in species interaction, community structure, and the dynamics of energy transfer. And that lets us ask questions that will be helpful in the future about things like how resilient a system is to disturbance. So how well connected a food web is can tell you about what will happen if stress is put onto that system. It can tell us about, as mentioned, how those systems link to each other, and then how things like nutrients or contaminants are cycling in a system. So this, as I mentioned, builds off of previous work. And I said Allison Jackson's name because she and I actually went to graduate school together in Oregon, but she also had worked in Maine. So when I had the opportunity to start thinking about moving here and building a research program, I called up Allison and said, you know, I'm reading about this program. I know you've been in Maine. Tell me about it. Is this a great place to work? I see there's potential for some really cool questions. I think they'd benefit a lot if we work together. And she was like, yes, it's great. I had so much fun. And we should do something. We had very similar system experience. So she actually, when we were in graduate school, worked in a different reservoir that was near the reservoirs that I worked in, but looked at songbirds, right, and mercury, and I worked in the water. And so the two of those, although they sound like really different disciplines, are really intricately linked because a lot of riparian birds are feeding off of emergent aquatic insects that spend their entire, young lives developing in an aquatic system. And so even though we work in different fields, our systems are intricately linked, and it was a great opportunity for us to work together. So we started to think about how aquatic and terrestrial systems might shift depending on community composition. This is a diagram that's an example from a paper by Colden Baxter and others. And the example here is that trout were put into streams that had native char. Those trout then ate the terrestrial insects that otherwise would have fed the char. The char focused more on aquatic insects, with the implication that then you don't see as strong of a linkage between those emergent insects and the terrestrial system, because the insects are being eaten, intercepted, before they're able to emerge and feed that terrestrial system. And so we wanted to see how this played out in Acadia National Park using carbon and nitrogen stable isotopes, and I'll talk about what those are, because to some people that might make a lot of sense, and to others it might not. We also wanted to sample birds to determine how much the aquatic subsidy use changed and whether that is related to what we're seeing in the different lakes, and then to kind of tie it all together. So I mentioned I was going to talk about DASIN, and that's because DASIN Weedup started in September of 2021 and has really focused on this question as part of his master's. So here's an example of kind of what we expect to find and what his thesis will focus on, and that's the differences in sort of aquatic communities. What to take away from this is that you can see when a new group of fishes are introduced, they can shift the kind of pressure, the strength of those arrows on different parts of the community. So in this case, you'd see more pressure on zooplankton, for example, and that could change what's coming out. The way you make those figures is through isotope sampling. Isotope sampling has advantages and disadvantages. One of the great advantages is that it's integrative. So you are what you eat, but not just what you ate last night. So you're not just having a fish regurgitate into a cup and then looking at what's there. because that might not tell you where it's actually getting its energy from. Isotopes are great because they can tell you where the material originated. And that partially has to do with how plants cause changes in the signature of carbon. And they can tell you trophic level. So how many steps did you take up from like a source plant material? And they also tell you how varied diets are. So if we examined everyone in this Zoom room, for example, we might be able to tell how spread out we are on a landscape, because there's also landscape patterns, but also whether some of us might be really omnivorous, some folks might eat a lot more meat, some folks might be vegetarian, and you can get an idea then of the niche breadth. So how diverse the different diets are for even species that are in, like, members of the same species within a community. So to give you a refresher of what this all means, I'll just do some little comics for carbon, nitrogen. And for carbon, as I mentioned, what we're looking at are these delta values. So basically, there are versions of carbon that are a little bit heavier than the most common carbon. So carbon 13 is heavier than carbon 12, and it's stable, so it's not radioactive. And then because of that, we can use it to figure out what's going on. And that's because the way that plants respire, sometimes they'll hold more of that heavy carbon and sometimes they'll hold less. And so you get this nice kind of biplot of where something got energy from and how far up it went. So the where something got energy from typically looks like this. More enriched things move to the right. It just means heavier. So for example, if you had someone eating fish and they were right in the middle there because fish were eating, say, aquatic vegetation, or insects that ate aquatic vegetation, and then you have an anteater that's eating ants, that's originally deriving its energy from terrestrial vegetation, you might be able to identify then someone who had a diet that's mixed between ants and fish. Nitrogen is a tracer instead of trophic source, of trophic position. So it works more like I mentioned mercury, where you get more and more of that heavy nitrogen as you move up. And that's just has to do with the fact that it's easier to excrete the lighter isotope. And so there's a saying, you are what you eat plus 3.4. It's not always exactly that number, but essentially the number gets higher as something eats the level below it. This is an example of kind of how those biplots and those arrow diagrams that I showed you earlier play out. So those arrows at the bottom can be derived from the plots on the top, which compare the trophic source, carbon, and the trophic level, nitrogen. And you can see in this example, these hypothetical fish are integrating both terrestrial source potentially of carbon and an aquatic source of carbon, and then merging upward as they consume more material. And then in this case, this is an example of what's pretty commonly seen and what we would not be surprised to see. And that's that when you introduce additional species into a system, what you can see is more niche specialization. So fish try to minimize that competition, which you do see more competition often if there are more overlapping taxa. They'll try to minimize that by basically specializing their diet. So you get much more restricted niche space, which is those circles, for taxa that overlap. And that's related to processes, right? So basically what you're eating will translate into how much mercury you accumulate, what's coming out of that system, how the system responds to something like disturbance or pressures or climate change. And in order to do this, we need all those pieces. So as was mentioned, we were out sampling. What were we doing? In order to get that information, in order to build those plots, in order to measure the carbon, nitrogen, stable isotope values, we need to catch fish. And then we take a lower caudal fin clip. We need to catch benthic macroinvertebrates. So we're out there kick netting and scooping up bugs. We need to catch zooplankton. So we're out there with a plankton net, towing around for zooplankton, to look at plants. So aquatic plants and terrestrial plants, to try to get an idea of what those sources are going to look like, so that when we watch carbon moving through the system, we can see kind of where the majority of energy is coming from, and whether it's produced in the lake itself or whether it's coming actually from the habitat surrounding the lake. And then, of course, these are the aquatic sampling components, which are what I've been focusing on in this description, and what I tend to focus on because that's my kind of specialty. There's also the terrestrial components, which we were able to sample thanks to Allison. So she came out as well and worked on sampling birds and terrestrial insects, as well as emergent insects, which are that kind of interface, right, between the aquatic world and the terrestrial world. And then right now, I wish I could show you lots of biplots, but stable isotopes actually take a fair amount of time. We've got them all identified and they're dried, and now we have to grind them and pack them into little tiny metal tins that then are combusted, and basically they have to turn a corner. And if they turn slow, then they're the heavier isotope, and if they turn faster, they're the lighter isotope. So they get sent into a lab for all of that to happen, and then we get back those beautiful data points, and we can make those plots, and we can talk about how the fish in the different lakes then are occupying different niche spaces or have different variability in their diets. These are examples of the actual samples as they are now, and some of the really neat aquatic insects and others that we can see. So what do we expect to be able to do with this? You might already have taken away baseline data. So I don't have a lot of stable isotope data for Maine. Each place can have a different, as I mentioned, kind of a locational signature. There's also a lot of overlapping fish taxa. So a lot of those fishes that are introduced into lakes in Acadia are introduced to lakes all over, because a lot of fish that get moved around are commonly popular game fishes, and they can behave very differently in different circumstances. So for example, when we look at bluegill in Oregon, which are an introduced species in Oregon, they're almost exclusively invertebrous in the reservoirs that I have experience working in. That's not always the case throughout the range in other systems. And so getting an idea of kind of what's normal here will be really helpful as we move forward and think about what these food webs look like in other places. It'll help us interpret existing information. So in some cases, for example, songbird mercury might not tie well to the mercury of, for example, dragonflies. And one reason why that could happen is because that terrestrial to aquatic link, or aquatic to terrestrial linkage, is not as strong. And that could be because of the fish that are there and the pressures that they're putting onto those aquatic benthic macroinvertebrates. And then understanding how fish assemblages impact feeding can help you to do things like this paper here, which is a great example of how you can use resource use to examine what will happen when different fish are interacting under novel circumstances. So to kind of project forward and see maybe where there are places of concern or overlapping taxa of concern. So in other words, Acadia lakes can serve as baselines for other lakes in Maine. Many of the species that are in these lakes are represented across the state. The current food webs are a great point of comparison for future conditions. And it would have been really cool, for example, if we had had food web information from before, yellow perch were introduced, for example, because they very likely did some restructuring, and we might be able to get at that by our comparison of lakes, but there's tremendous advantage, right, to being able to track these kinds of things over time and increasing our interpretive power over other components like mercury. So that's what I have prepared.
That's great. Thank you so much, Chrissy. Excellent presentation, and it's such a useful reminder for me of how isotopes work. And I actually have a methodological question, but let me just remind everybody that if you have questions or comments, this is the time to air them. Feel free to unmute and just ask your question. We don't have too many people on here. I don't think I need to moderate too much. Although Stephanie, all right, you've raised your hand digitally, so go right ahead, Steph.
Okay, well, this is kind of a stupid fish
I need
psychology
to
question.
moderate too much. Although Stephanie, all right, you've raised your hand digitally, so go.
Yeah, so we are just wondering, do fish ever choose what they eat, or do they eat anything that presents itself in front of them?
That's a great question. So part of this I'm going to answer based on previous stable isotope and actually an economics theory, which is you focus on what you think you can find. So what we found is if fish densities seemed to be low enough, that fish that would typically look for other fish will start just seeking out and consuming invertebrate prey. So they probably do eat a fish if they come across it. But I think a great analogy is what people do. And that's if you're in a new city and you really wanted, for example, a hamburger, you might spend a while looking for it. But if you just can't reliably find what you want to eat, you're going to pick a new focus, right? And you're just going to say, that's fine. I'm going to eat there. That's the new place I eat. That's what I eat. And I think fish, as far as we've been able to tell from previous studies, do something similar, which is that they're going to pick a prey item that they can reliably find, because you can't just be reacting. I mean, you could be, and trying to swallow everything. It wouldn't necessarily be the most efficient. And you might be one of the fish that does nothing but eats pollen and then starves to death. So.
Stephanie, I have years of anecdotal evidence that trout do not eat what I present to them. So they seem to be very active and finicky eaters. Chris, you have a methodological question. And I don't know if this is significant at all, but as we increasingly burn fossil fuels and change carbon isotopes in the atmosphere, does that change like calibration curves or other aspects of methodology for this sort of work?
I mean, typically when you're thinking about dating with carbon, like carbon dating, you're using atomic, like nuclear weapons decay over like long time scales. But yes, they're probably, so even within a system like a lake, part of the reason that we did a BioBlitz when we were out there is to get all the isotope data from the same point in time. So even across, say, June, July, August, the carbon signatures can start to shift, especially for the production that's happening within a lake itself. That's been observed in other places. So there definitely is a component, not necessarily related to human activity. I think that might be on a finer scale than we would have a great way of measuring, because isotopes are so integrative. But there certainly are patterns where you see shifts in baseline values, even across seasons within a site. So certainly keeping in mind that these aren't super stable targets as these studies go on over time is really important. And your example for fish thinking of specializing is great, because I think that's another example of fly fisher folks making sure that they pick kind of what is hatching, because the fish are getting a search image and really focused in usually on that particular taxa.
And other people on this call who have deep interest in water and water quality and fish and other wildlife. So I'm hoping folks have questions, and feel free to speak up directly or raise your hand digitally, whatever works for you. Questions, comments, reactions?
Hey, this is Kate Miller. I don't know how to raise my hand on Zoom, so I'm just going to turn my video on. One question I had is, did you do anything with aquatic plants, or is their signature so obvious that, okay?
No, that was one of the kind of represented by a baggie and those little, but yeah, we collect phytoplankton and the emergent aquatic vegetation. It can be pretty different, actually. The littoral vegetation can be different from the pelagic phytoplankton production, and that's different from the terrestrial. And then each of the different terrestrial plants, you know, can have a different signature. So, which for folks who are interested in stabilized isotopes and haven't read it, there's some great work by Renee Brooks and others looking at how to identify adulterated beer with stable isotopes because of the different plant signatures. So barley and corn have really, really different stable isotope signatures. So yeah, capturing the different vegetation types, especially if you're talking about grasses or something versus phytoplankton versus a tree, tremendously important.
Great. Thank
Yeah, well, hopefully you guys can have me back in like a year, maybe half a year.
Yeah.
We're gonna get through it really fast, and then we can look at actually what the food webs look like. Although that can take a lot of time to tease apart in a presentation.
Would like, I don't know the full scope and reach of stable isotope analyses and what you can infer from them, but can they tell you anything about when a fish invasion occurred? No, okay.
No, because really, I mean, not the way we're using them. So we're using fin tissue. It's pretty fast turnover. And really, it should give us an idea of what fish have been eating, depending on the growth rate of the fish for the past few weeks to month or so. But no, what could help is, you know, monitoring over time, even if you weren't capturing directly something like that, some kind of disturbance event. If you had, say, if we went back and sampled isotopes in 5 years and some of the taxa look really different, then that would tell you that, you know, something might be going on there. And it would be worth exploring whether the community has shifted or some component of the environment is really different.
And then just sort of referring to your very first couple of slides and introducing what the Cooperative Research Studies Unit is like. I think you used the term client or other partners, and so cooperator. Okay, so I'm thinking, you know, at some level you're doing research that your cooperators need done. This project you described as, I think it's coming from you, right? But and I don't know the history here, so I don't know if Acadia said, "Hey, we really need to know more about fish communities and what happens when they change and that sort of thing." But are there, and I realize you're not using the term client, but if there are sort of client organizations out there who are interested in this kind of thing, do they reach out to you and say, you know, we want to know more about fish communities or X, Y, and Z that can be determined through stable isotopes, and then you respond to that, or how? What's the relationship?
No, so the term cooperators is born, I think, out of the fact that, again, the cooperative research units, I think, are often affiliated with land grant institutions. So it's kind of harkening back to the days of, we have a lot of, you know, fish and wildlife. Many of them are popular for various resource needs, and how do we get answers to manage them? As far as our actual research, we function a lot like university faculty, in fact, almost exactly. But it is important. So at the cooperators meeting, we had one last week. The projects that we conduct are basically approved by the partners. So for example, if I were doing something extremely esoteric, that might be an issue, but really a project like this, and I think stable isotope data in general have tremendous benefits for management potential questions of interest. So approaching these kinds of kind of broad topics, sometimes it takes some time and some steps, but these are how you get answers to how do you better ensure that the species or environments that you care about are there 30 years from now.
I came a little bit late, but I do have a question. Thank you for the presentation, quite interesting, and I look forward to learning more as your results come in. And I don't know anything about stable isotopes, but is it possible to use historic specimens that might be in jars to look at food webs at an earlier point in history? So a lot of people did collecting at some point, and I just didn't know if the pickling, in a sense, or preservation techniques might ruin the samples you need to do your analysis.
They certainly don't ruin them, but they don't make it easy. And part of that is a lot of what carbon and nitrogen, at least in particular, is sometimes like those lipids and things can be extracted, for example, by things like ethanol preservation. And so there are folks who have spent a very long time coming up with correction factors, but they're not always consistent. And the length of time that a specimen was preserved or how it was preserved can impact that. And so using information from collections can be really challenging, especially if you don't have all the pieces of the puzzle from that time. So for example, like we talked about, carbon might not be stable all the time. And so even if you could get a perfect signature, you might not know where it fit within that kind of network that you would need to put it together. But certainly fish collections offer, tremendous benefits, not necessarily easily in the case of isotopes, but certainly in the case of, what did the fish community look like? Things like fish diseases. Yeah.
And insect collections are generally dried. Now there may be arsenic, but if you took a leg or something or an antenna, could that be used? I'm just
yeah, for sure. For insects, it's a different story. And if they're just dried and they've been dermestids or others haven't gotten to them, then yes, you certainly could use especially terrestrial insect collections and get some really good stable isotope information.
Well, and as you probably know, Acadia has a tremendous historic resource in the museum of historic specimens from Proctor's era and after that at some point could be used to maybe make some comparisons to what we're seeing today. So anyway, thank you very much.
Yeah, that would be really interesting. I think you'd be talking more terrestrial insects, but maybe you could look at changes in vegetation, how that impacted
well, there could be some aquatic insects that were kept, and I don't know if they would have been dried as well, and pinned.
Yeah.
Like beetles, things like that. But anyway, just wondering. Thank you.
Rebecca.
Hi, I thought David was going to ask a different question, so I'm going to just bring it up. as you know, our lakes are somewhat contested spaces in terms of jurisdiction and authority and management. And certainly part of the interest in your work is because of the opportunity to open up some of that dialogue with others. And I know you, when you were out in the field, that you had other folks with you or were helping, assisting you in this work. Can you speak a little bit about the potential for opening dialogue around using your work as another way of understanding some of these impacts that have gone on in the lakes, towards more conversations around management, maybe?
Yeah, sure.
I'm dancing around a bunch of things, but we all know what we're talking about here.
Yeah. And I mean, that's so when, so the Acadia National Park, this is actually hit on in some of the previous work as well, where as a national park, it's really unique in, I think it's none of the lakes have the entire border within like completely uncontested national park space, as far as, especially when you consider the boundaries of the watersheds. And so there are, even for example, Little Long Pond, which is not part of our study, and it's outside of the park boundaries, but the headwaters, like the streams that feed into it, are within the park. And so you hit this kind of mismatch of streams coming in that are outside of the park, lakes that are in the park, half the lakes in the park, and then also with different histories for management. So the state is managing with different objectives potentially than the National Park Service, or them like a local municipality or water district. And in the case of this project, for example, IFW has been a really great partner in helping us to collect the fish samples, because they're also managing the fish communities. And so not necessarily with the same objectives as a National Park Service, because part of their mandate, right, would be to provide recreational angling, which some taxa are more valued than others. From that perspective, I think there's tremendous benefit to understanding how a fish community functions, right, regardless of what your specific objectives are, because it lets you better manage for different outcomes. And so I think we can all get to the table and agree that in general, most people like having, you know, functioning ecosystems and fish that are growing. And, you know, also there's definitely overlap in not wanting new taxa to find their way into these systems. And that encompasses kind of people management, which can mean managing the fish to manage the enthusiastic folks who might have a favorite species that they can't find in their local water bodies. So knowing how fish communities work, I think will benefit all of us, and is a great way for us to just meet at the table and just start talking about who's there and why it matters. So, for example, in a system where you might be, you know, augmenting the populations of some species, you know, how does that impact other species? And then, you know, how are they connected, or all of that will come together. But yes, I think we do need to have a conversation, ongoing conversation, and this has been a big part of that. Sorry, that also kind of dances around the we all love the fish issue, just in different ways and different fish.
Good questions, Rebecca and David both. Other comments, reactions, questions, etc. for Chrissy? Were abundances pretty similar across the lakes? Where are the fish? That's what I want to know.
Yeah, no, actually, it's really interesting because that was one of the things that surprised us. And you might be touching on some of our like, oh, this is weird. Because in some systems, both lakes might have shiners, for example, and in one place, that's all you're catching, it feels like. And then the other taxa feel relatively rare. And then you go out where the compositions should be pretty similar, and you'll get, 2 shiner or 2 suckers, and then everything else is much more abundant relative to that. So I think there's certainly some really interesting dynamics going on in some of the systems for what the different dominant taxa are. The lakes themselves seemed, in some cases, pretty different as far as their productivity. And part of that might have to do with how heavily they're being, hit by fish eating the invertebrates, for example. So in some systems, we had a much, much harder time getting large numbers of aquatic macroinvertebrates than in others. So, yeah, and not just it seemed as a function of substrate, but.
So that pronounced variation among lakes in the abundance of different species of fish, and so the sort of community structure, you don't see that kind of variation in other landscapes that you see here, and what? So do you know why? Do you have any thoughts on why it's weird here?
I mean, I think it's partially because even though the compositions are so similar, they are different, and those differences are enough to potentially produce some pretty different dynamics. I mean, I think we'll see. That's my current hypothesis. But yeah, for example, we worked in reservoir systems, the communities would be different by, a few taxa, typically fish taxa, but the structure was relatively similar. The fish that were abundant were always kind of the same species, but part of that too might be the lakes and how they're kind of sizes. Like, part of it might be the who's there, part of it might be the environment that they're playing out in, and I think we'll get a better idea of that when we're able to see how they really are interconnected, and whether we see those kind of same connections across different systems. And then you'd think maybe it's more environmentally driven, or whether there's, one taxa is there and then suddenly we see these consistent shifts where that taxa is causing all these others to switch what they're eating to something different. So I think we'll be able to start getting at that question, but maybe not all the way there.
I did want to ask, did you access any of the historical information, and just in terms of the previous fish inventory work that was done in the park, and IF&W, stocking records, things like that, just to help kind of piece together the different parts?
Yeah. Well, yes, we had to, because I didn't know what we were going to catch, so that we could get, permission to touch the different fish that are in the different lakes. And then also, so we went back to those historical reports. We looked at the different stocking regimes, and we looked at angler reports. So that's how you can see. So we knew we were likely to catch yellow perch, in addition to the taxa that have been previously recorded, for example, because you can see anglers posting photos of it on some of the popular media sites. So for example, Dason, the graduate student who's leading a lot of this work, pulled up FishBrain, which is like a fun little phone app that a lot of the anglers are using, and you can see they'll post photos of their fish, which helps too, because then you can verify the ID, but you can see potentially as species that were not previously recorded in a system.
Did you look at Upper Hadlock at all? I wasn't, I wasn't in your talk at the beginning, so I didn't know if you looked at that lake or not.
No, right now, this is like a small kind of pilot effort, right? So we've got 5 lakes, which were Long Pond, Jordan Pond, and Betty, Lakewood, and what am I missing? Take it back.
That's okay. I was interested in Eagle Lake, Big Six.
Yes, and Eagle Lake.
I was just interested in Upper Hadlock because during my tenure at the park, there was a one-time introduction of splake that was in effect a mistake. We had assurances from IF&W they wouldn't do it, and somehow the message from the director, and somehow the message never got to the hatchery. So anyway, just that might be a unique, whether or not you could tell. And we know when that happened, whether you could tell any changes in fish, in the food webs based on that one-time introduction. And, I don't know if the fish survived or whether they were caught out, but anyway, that's why I asked that question.
Yeah, well, if it's been 10 years, they've probably cycled out. And actually, a fair number of these lakes have histories like that. I mean, they got very creative 100 years ago with what anglers might be interested in. But many of those introductions didn't take. And I think, we've kind of entered a new era, right? Not just in Maine, but just generally nationally, where state agencies no longer typically introduce fishes that weren't there. In fact, so IF&W still does some stocking, and it's focused on cold water taxa with the idea that, you're augmenting closer to a native fish, or at least a fish that's native to Maine, and then hopefully preventing introductions of what might be more detrimental taxa. So fish that, for example, folks in Oregon love moving around largemouth bass, love, moving around some of those big game fishes that actually, even while we were out sampling, people were asking me where to catch some very interesting fish taxa that I would maybe not be looking for as an angler in northern New England, but
Thank you.
Snakeheads, right?
yeah, no, it's interesting. People, and we all love what we love, and you get used to something, and it can be really hard to leave it behind.
That's a great note to end on, Chrissy. And we're at the top of the hour, so we should end. But Chrissy, thank you so much for the presentation, spending your time with us. It was great. Thanks to everybody here for attending. Just a reminder that this recording will live on the Scudic Institute Facebook page. And somebody put a chat right in the in the message box there from Bob Bechtold. So look for the presentation on the Scudic YouTube channel. And Chrissy, hope we can hear from you next year and hear about your results. So
yeah,
thanks.
looking forward to it. Thanks, everyone.
All right. Bye-bye.